Hello, my lovely anatomists and physiologists. Michele Glass here, back with chapter 12. We are getting ready to talk more details about that action potential, which we've mentioned before is like the signaling, the electricity from the nervous system, right? And before we really get into that particular process, I wanted to do a few reminders. So when we talk about our cell membrane, remember this is going to regulate the entry and exit of materials into the cell, right? So the cell membrane is regulating the entry and the exit for materials getting into the cell. We can also talk about it as our cell membrane as that phospholipid bilayer, right? And so remember, this means only lipid soluble materials, chemicals can actually cross through that phospholipid bilayer. So what that means is if you're an ion, if you're charged, you're going to have to use a protein channel or a protein carrier in order to get into that cell membrane, right? And so that takes us right to this particular conversation about those ion channels. So remember, ion channels are made of proteins, and protein shape determines function. And so proteins, because of this, can be really, really particular, really specific. And so what we'll see is that it's going to be charge specific, meaning only a cation or an anion is going to be able to enter. So you can't have a channel that is allowing for both cations and anions to go through that same channel or doorway. It's going to be either a cation channel or an anion channel. Remember, your cation is your positively charged ion and your anion is your negatively charged ion. It's also going to be diameter, or what we could say as size specific. So because of the nature of the channel, it's only going to either allow positively charged ions or it's going to allow negatively charged ions. Because of the size of the channel, that's going to further determine, you know, who's able to actually really cross. So we have, you know, a high level of specificity when we talk about ion channels. It's not just like a random doorway that anybody can use, right? And then we also talked about they could be gated. And if they're gated, I like to use the idea of the gate can like close or open. When we say close, we should really think of like deactivate. And when we say open, we should really think of like activate. So we're going to see there's actually several different types of gated channels. We're going to talk about 3 examples. So we have what's called the ligand gated channel. And this is what we've been talking about as the chemically gated. The ligand is saying a chemical has to bind. So it's just a really more specific terminology. It's telling us the same kind of thing. So in the case of a ligand gated or a chemically gated channel, a chemical has to bind. That's what we saw in chapter 10 when we looked at acetylcholine binding to that channel on the motor end plate. We can also get what are called mechanically gated. And this is where pressure, physical mechanical pressure, will actually activate or deactivate, open or close. For me, I think about there used to be these little plastic coin purses, and when you weren't touching them, they were closed. And if you squeezed them at a certain angle, then the coin purse opened up and you were able to get into your coins, your money. So I always think about that when I think about a mechanically gated channel. And then the last of these is the voltage gated. And we saw these also in our chapter 10. And the voltage gated, remember, was the change of charge, which really just means movement of ions. And that's going to either open or close, activate or deactivate the channel. Just to mention here, we also have what are called leak channels. And these are more of like a random, they're either always open and allowing the movement of material through, or they have a randomness to when they open up and close. So it's not being controlled in the way that a gated channel is controlled. The last thing for us to remember is our old friend, sodium potassium exchange pump. And remember the significance here. I'm going to draw my cell as a circle, and I'm going to draw potassium inside the cell as like a real big, taking up all the space. And I'm going to draw a really huge sodium outside of the cell to represent we have been actively pumping out 3 sodium and actively pumping in 2 potassium into the cell. So the sodium potassium exchange pump, remember, is pumping 3 sodium out of the cell, 2 potassium into the cell. And so this is doing like 2 things for us. And let me change color so it pops a little bit better. Sodium potassium exchange pump is doing 2 things for us. It's setting up a high sodium concentration in the ECF. So here I've used my up arrow to say high. I've used my brackets for concentration, and I use my chemical symbol for sodium. ECF, remember, is your extracellular fluid. So we have concentrated a whole hell of a lot of sodium into the extracellular fluid. We've done the reverse of that with potassium. So we've concentrated the potassium inside the cell. So this is important. And this has set up a resting membrane potential where the ECF has a positive charge and the ICF has a negative charge. So when we talk about resting membrane potential, we're really referring to the cell membrane itself. And the nature of the sodium potassium pump, pumping all of the sodium on the outside of the cell, is setting up this potential because you have such a high positive charge in your extracellular fluid. Any ions, any anions, negatively charged ions, any negatively charged proteins are hanging out right inside that cell membrane. So I'm going to draw some negative symbols on the inside of this cell that I drew over here to kind of remember when we measure across that cell membrane, we're going to get a negative charge on the inside. And that negative charge on the inside, that setup of these ions in different places, is making what we call the resting membrane potential. So that's going to be huge as we progress in our discussions. And when we talk about a cell at rest, what are we saying? We're going to say that the gated channels are going to be closed, or we could say inactive. We're going to say that sodium ion is 10 times greater in the ECF than in the ICF. So this is a significant difference. Let's remind ourselves that the potassium ion is going to be at a greater concentration in the ICF, so inside of the cell. But remember that our cytosol, all the fluid inside of the cell, has lots of anions and negatively charged proteins. And so this gives us a resting charge of negative 70 millivolts. So this is our resting, we call this our resting membrane potential. And we're memorizing this number. So anytime we talk about a cell at rest, we should understand its resting membrane potential is negative 70 millivolts. And it's negative 70 millivolts, it's negative inside because of that sodium potassium pump, creating 10 times more sodium in the extracellular fluid compared to the intracellular fluid, because those other gated channels are closed, and so forth. When we talk about membrane potential, just this kind of term, membrane potential, what we're talking about is the charge across the cell membrane. Okay, we're measuring it in millivolts. So we can do little m and then big V. And then we're comparing, it's a comparison. We're comparing the ICF to the ECF. So because all that sodium is in extracellular fluid, it's way more positive compared to the intracellular fluid. And so that's how we're getting that negative charge. Last thing to look at together, we have a couple of important terms. So we got to make sure that we're keeping up with the differences here between depolarization, repolarization, and hyperpolarization. Just over here to the edge, when we think about polarity, remember we have opposite ends. And here we're looking at across the membrane. So in the case of depolarization, the D prefix means to remove. So in depolarization, you're making the cell, and here if you wanted to say ICF, more positive, like the ECF. And so this is happening because you have movement of sodium into the cell. Sometimes it's calcium ion. So in the case of depolarization, you have this positively charged ion moving into the cell, making the inside look more like the outside. So we call that depolarization. If you repolarize your cell, you're going to return to resting membrane potential, which remember we've defined as negative 70 millivolts. So in the case of repolarization, you're going to return to resting. So you're going to make it more, we could say more negative or less positive. So you're heading toward the negative 70. This is happening when potassium ion leaves the cell. So when potassium ion is leaving the cell, that's repolarization. Sometimes we'll see chloride ion can enter the cell. Sometimes we'll see that. Repolarization just is making the cell more negative. In the case of hyperpolarization, you're making that polarity, that difference across the membrane more extreme. So here in the case of repolarization, this is caused by continued movement of potassium out of the cell, making the cell more negative than resting, which is negative 70 millivolts. So depolarization is to remove the polarity to make the inside look like the outside, and we do that by allowing sodium to move in. Repolarization is to kind of reestablish our resting membrane potential, which is negative 70 millivolts. So this occurs by having the potassium leave the cell. And then we have a situation called hyperpolarization, where that potassium just keeps leaving. And this actually makes the cell more negative than how we started. So we're even more negative than negative 70 millivolts. Okay. Stay tuned next for the story of action and potential. And as always, take care of yourselves and each other.